All right, let's go ahead and get started. So we left off on chapter 42, which is gas exchange and circulation. And we had noted that there were two kinds of circulatory systems, open and closed, right? And if you have an open circulatory system, like lots of arthropods, lots of crustaceans, most mollusks except for cephalopods, right? Then it doesn't, the circulatory fluid doesn't get called blood, it gets called hemolymph. And in many cases, but not necessarily all cases, it is separate from gas exchange, right? So in hemolymph, in, for example, most insects, it doesn't have any sort of pigmented molecule that binds to oxygen at all, right? Which is why when you often step on bugs, it's just kind of white and gross. And again, it's because hemolymph usually isn't involved in gas exchange. There are exceptions, of course, because this is biology. And there's always exceptions. So for example, spiders, right, are arthropods, and a lot of them have pigmented. Yeah, so it's in an open circulatory system, which is kind of weird. So anyway, meanwhile, in lots of other organisms, notably vertebrates, as well as most annelids, like earthworms here, you have a closed circulatory system and cephalopods. And that's where blood is confined to vessels. It's distinct from the interstitial fluid. So there is interstitial fluid, right, that's sort of bathing all of the cells and tissues, but it's distinct from blood. Okay, so as you might imagine, right, in both cases, in an open or a closed circulatory system, you have some kind of heart organ that's contracting, that's providing some sort of locomotion for the fluid. But it's more efficient in closed circulatory system just because you have less volume you have to pump, because you don't have to pump through an entire body cavity. You just have to pump through the vascular tissue. Okay. Okay, and I think we did touch on this also last week, right? We have sort of three different kinds of blood vessels. We've got arteries, capillaries, and veins. Arteries start out, right, from the heart going out, start out as arteries. They branch into smaller arterioles, and eventually those branch into capillaries, right? When you have huge networks of really thin capillaries, right, that gets referred to as a capillary bed, and that's the site of gas and chemical exchange between blood and interstitial fluid in organisms that have closed circulatory systems. Coming from the other end of those capillaries, right, so now gas exchange has happened. Those capillaries converge into venules, which converge themselves into veins, which ultimately return blood to the heart. Okay. So we're going to look at some circulatory systems now, but we're going to confine it largely to vertebrates, just because it's already complex enough, and there's already enough diversity among vertebrates. But suffice it to say, stuff gets pretty weird when you look at other organisms with closed circulatory systems. So for example, if you look at octopuses, octopuses have several hearts. Yeah, and it's complicated. So luckily for us, vertebrate hearts, there's usually just one. I can't think of a time when there's not. But the number of chambers in the heart will vary by taxon, right, by different groups. So for example, in most bony fishes, as well as cartilaginous fishes, sharks and rays, you have a two-chambered heart. Okay, always, 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 no matter how many chambers of hearts you have, blood returning to the heart goes into an atrium. Okay, and blood leaving a heart goes through a ventricle. Okay, so bony fishes, sharks, rays, all have what's called single circulation, right? So one contraction of the heart muscle, right, pushes blood from the ventricle through the gill capillaries, right? So that's a capillary bed that exchanges CO2 with oxygen. So now you have oxygen-rich blood flowing into another capillary bed that is the rest of the body, right? So this is where all of the exchange happens in the interstitial fluid of the body of the organism, right? So that's called systemic circulation. And this, in the case of fish, is called gill circulation. Okay, so there's one pump of the heart that's doing both of these circulations. Okay. So hold that thought. We'll come back to that. In all other vertebrate taxa, right, amphibians, reptiles, mammals, we have double circulation. Okay, so in all of these taxa, you now have a left atrium and a right atrium. Okay, so note, this is from the organism's point of view. So this is the left, even though to you looking at it, it looks like the right. Right, so this is always like pretend you're the animal, and that's the left. Okay, so we've got a left atrium and a right atrium, left atrium and a right atrium. Okay, but in amphibians, we have just one ventricle. Okay, so we don't have a four-chambered heart. It's actually a three-chambered heart. Why don't we start? Let's start with the left ventricle. Okay, so the left ventricle, we're pumping oxygen-rich blood, right, through the systemic circuit. Okay. So oxygen-rich blood is being pumped through. It's exchanging with tissues in the body, right? The system, right? All of the other organs, tissues, et cetera, in the organism. Okay, delivering oxygen to them, taking up waste and CO2 from them, and pumping that into the right atrium of the heart. Okay, which then flows into the ventricle. Okay, which then pumps through the arteries. We'll talk about this in a little bit more detail in a second, right? But once it goes through the ventricle, it can take a different circuit, the pulmocutaneous circuit in the case of amphibians. Okay, so here is where you have oxygen-poor blood now exchanging with oxygen-rich air. Okay. So there are actually two different circuits. Does that make sense? Okay, so that's why it's called double circulation. Okay, so let's take a vote. Do you think blood pressure is greater in double circulation than it is in single circulation? So give me a 1 for yes, you think this statement is true, and a 2 for no. There were a lot of abbreviations there. Do you want me to write it out? Does it make sense? Okay, wow, divided group. So 1, let's get a representative vote from 1 if you don't mind.
I don't have a good reason for
Okay. Does anybody who voted 1 have a good reason? Yeah, Carlos.
I just think you just need more pressure to like to feel to feel those capillaries, you know.
Okay.
That's really just the main reason.
Okay. That's definitely a good guess. And then 2, what's someone who thinks they have a good reason for 2?
I had a good reason, and I realized it wasn't consistent.
Okay, well, here, let's take a look at this. So let me just look at the logic of this for a second. Yeah, I'm with the 1, so blood pressure should be higher for double circulation than for single circulation. So let's look at single circulation here for a second. Anytime you're going from an artery into a bunch of capillaries, right? It seems like that should be increasing blood pressure and speed. So think about it like if you have a garden hose in your backyard, right, and you close a little bit of that space, the water coming out of it is now coming out faster, right, with higher pressure. So you would think that that's what would be happening here, but you'd be wrong. So the reason that you're wrong, not that you personally are wrong, this hypothetical you is wrong, is because there are so many capillaries, right? The total sort of diameter, if you were to cluster them all up, is greater than the artery that's leading to them. Does that make sense, right? So you're actually emptying out into more volume once you spread out across these capillaries. Is this making some kind of sense? Okay. So as an example, there was a group in lab last week where you guys were, I think it was your group. You guys were trying to figure out the splitter, the little ventilation splitter, right? So if you've ever had to like owned an aquarium or something, right? If you have an air pump, so this is an air pump. Okay. If I'm pumping air into an aquarium, into one air stone, that's how you oxygenate your aquarium tank, by the way, okay, then you get a lot of pressure from this one tube, right? But if I were to take this tube and connect it to a splitter, each one of these is actually going to have lower pressure, right? Because in total, I'm increasing the diameter, or the circumference that air is going through, 3-fold. Does that make sense? Okay, so it's as if I'm going from a tube this small to a tube 3 times the diameter. So the water pressure is going to be a lot lower there. Did I just make it worse? Okay. Carlos.
I had a great conversation with
Melissa?
my daughter's classmates today. We were talking about why spiders, even though they have 8 eyes, they can't see. And he told me it's like if you had 2 cups with our 2 eyes, but then you had to share it amongst 8 cups. There's less water in each cup.
Okay. I'm not sure I agree with the premise of spiders not being able to see. So some spiders can see super well.
I mean, the idea of when you have to share it among all those, there's less.
Right.
Yeah.
Yeah. So same idea. So we're basically increasing the volume that this liquid has to go through. Okay. So it's as if we just have like lots of splitters. Okay. So what happens now is maybe blood pressure started out high, but now it has to go through all of this volume. Okay, so blood pressure goes down, and now it has to do it again. Okay, in the same single pump. Okay, so one pump of the heart now has to go through 2 capillary beds, whereas in double circulation, one pump of the heart takes you through 1 capillary bed, and then another pump of the heart takes you through the 2nd capillary bed. So overall, blood pressure is a lot higher in organisms that have double circulation. Does that make sense? Morgan?
That's why we do the 2, when you take blood pressure, there's the systolic and the...
We'll get to that. Diastolic?
Yeah,
Yeah, we'll get to that. There's
Is that kind of related, or am I off?
a little bit off, but not too far off. Alex, what are you thinking?
Well, I was just thinking about like doesn't it also have to do with like relative blood volume and stuff like that? And, like,
Totally, yeah. So there are a lot of things that affect blood pressure, right? The total volume going through, right? How dilated all of these capillaries are, right? So how vasoconstricted or vasodilated they are. Pumps per minute, like how, yeah. So there's a lot that goes into it, but in general, yeah. Okay, so looking at amphibians, the first thing that you notice here is in the pulmocutaneous circuit, that word pulmocutaneous. Can anyone sort of parse out some of the prefixes? Pulmo meaning probably lung. Yeah.
Doesn't cutaneous have to do with like surface?
Yeah, it's skin. Okay. So pulmocutaneous means that gas exchange in amphibians is happening both via their lungs and via their skin, okay? Which is kind of cool, kind of weird, right? So if you look at this ventricle, okay, there is nothing separating. Okay, well, there's a slight thing. There's a little bit of a ridge here in amphibian hearts, but it's not fully chambered. Okay, so there's not a full division here, but there's just like a little bit, a tiny little division here. Let me fact check that. I could be wrong about that. No, I'm right. Okay. So what ends up happening is amphibian hearts are pretty efficient at having oxygen-rich blood from the left atrium going up through this side of the artery, right? The left side of the artery versus the right. So about 90% of the oxygen-rich blood goes the correct way to get delivered to the body, okay? And the other 10% goes to get oxygen-rich again. It's already oxygen-rich. Whoops, womp womp, okay? So that seems really inefficient. That seems like a terrible idea, right? It seems like a really poorly designed heart for an amphibian. But here's the thing, is that sort of incomplete seal of that chamber actually allows amphibians to continue exchanging gas underwater through their skin, okay? The exact details of that are like a little bit unclear to me, and I've spent some time looking at it, and I don't want to mess it up thinking about it, but the words are on the PowerPoint. I won't ever ask you to explain in detail about that, but essentially they can shut off the exchange with the lungs and only continue exchange with the skin capillaries. Okay, and again, it's that incomplete seal between these two chambers that facilitates that. Okay, so in reptiles, they have the ventricle has this ridge that goes all the way, but as soon as blood exits the ventricle into the artery, it can take either of these routes. So again, there's a little bit of an allowance for some crosstalk here between the oxygen-rich and oxygen-poor routes. And again, this is because some reptiles, like crocodilians, can shut off gas exchange in their lungs and remain submerged for a while. Okay. To make things even weirder, right, you have some reptiles that breathe through their butts. This one does. Okay. right, so let's take a look at, so this is what mammalian and bird circulatory systems look like, right? So just like other terrestrial vertebrates, right, we have a double circulation, right? But unlike other reptiles, birds have four-chambered hearts, just like mammals do. Okay, so there is a complete separation, right, between pulmonary arteries and the aorta, right? So there are, it's not one artery that splits off. It's basically two separate arteries now. Okay. So oxygen poor. Blood circulates through the pulmonary circuit. Notice this is no longer the pulmocutaneous circuit, right? It's now just the pulmonary circuit. No more skin breathing here. And then oxygen-rich blood flows through the systemic circuit from the aorta. Okay. So why might this be adaptive for birds and mammals in particular? What else do birds and mammals share with each other that they don't share with other terrestrial vertebrates? Anyone remember? Close? Yes. Yeah, so they're both endothermic, okay? So that's a pretty high energy demanding lifestyle, which means you better be damn good at delivering oxygen to those cells, okay? So that's why it makes sense to have sort of complete separation, so there can be no mixing of oxygen poor and oxygen rich blood in the ventricle, okay? There's now two ventricles. With me. Okay. Does anybody have any questions so far? Good, good. Okay, so the left side of the heart, right, is participating in two circuits. The right side of the heart is participating in two circuits. Okay, the left side of the heart is dealing exclusively with oxygen-rich blood. The right side of the heart is dealing exclusively with oxygen-poor blood. Okay, so that's it for the first bullet point. We talked a little bit about different circulatory systems. Now we're going to focus pretty much exclusively on mammals, occasionally drawing comparisons between a couple of other organisms down here. But we'll talk about just coordinated cycles of heart contraction in mammals now. Okay, so we're going to just do a little tour of the circulatory system now. So obviously the need for oxygen is really high in mammals. Let's start out, let's see, where do they start out? Anybody know, without looking at the labels, I know there's tons of labels here, but you're going to have to know this anyway. So how do we, is this the right side or the left side? Right. Okay, is it a ventricle or an atrium? It's a ventricle, right? So the ventricles are on the bottom, the atria are on the top. The atria are where veins return to, okay? And the ventricles are where arteries come out of, okay? So we have the right ventricle here, okay? So remember the right side is dealing with oxygen poor, and this is, oh my God, an artery. Yes, that's the word for it. Okay, so this is an artery, and it's an oxygen-poor artery. In fact, it's the only oxygen-poor artery in the body. It's going to, yes, right, the pulmonary circuit. So what do you think this artery is called? Pulmonary artery, right? Okay, so it's going through the pulmonary artery, right, splitting bless you, going to the separate lungs, exchanging in the capillary beds of these lungs, now becoming oxygen rich. Okay, so now as we're returning to, what's this one? Left atrium. Yep. Okay, so we're in the left atrium now. So were these arteries or veins?
Veins.
Why?
Because they're returning.
Because they're returning to the heart. Okay, so these are pulmonary veins. So an oxygen-rich pulmonary vein. Okay, so now we are in the left atrium. Okay, blood goes from the left atrium. There is a contraction here that gets blood into the left ventricle. A contraction here that pumps blood from the left ventricle now into the systemic circuit. Okay, so it's taking half of the blood towards or some portion of the blood towards the top part of the body, and the other portion of blood towards the bottom part of the body. Okay, in either case, there's a big old capillary bed representing the whole body. Okay, gas is exchanged. Now we have CO2-rich and oxygen-poor blood returning to, what's this one? The right atrium. Okay, so are these veins or arteries? Veins. Okay, so this is called the superior vena cava, and this is the inferior vena cava, right? Superior because it's coming from above, inferior because it's coming from below. Okay, and both of those venae cavae in plural, okay, are entering back into the right atrium. Okay, pumps into the right ventricle, pumps into the pulmonary arteries again. Are we good? Okay, so I definitely would and do expect you to be able to name those chambers of the heart and just like generally describe how blood flows through those two circuits.
Not the superior vena cava and inferior vena cava, pulmonary artery, pulmonary vein. You don't expect us to know anything?
I mean, they're not technically on the learning objectives, but like
They're pretty easy, right?
they're pretty easy to.
Isn't it like 2D stuff?
Yeah, we'll get to that.
I learned a lot about the heart and the circulatory system because I was actually born with a couple congenital heart defects.
I bet you've learned a ton about it. Yeah.
I was born with partial anomalous pulmonary vein connection. I actually had open heart surgery to repair that back in 4th grade.
Wow. Wow. I definitely want to ask you some questions at some point. Okay, so right, we have a separation between atria and ventricles, okay, and there is in fact a valve between the atria and the ventricles on each side, okay. Those valves are called Atrioventricular valves, right, for unsurprising reasons, right? It's the valve between the atrium and the ventriculum. So you may have learned these as the mitral valve and the tricuspid valve. Okay, those are both AV valves. Okay, so that's one of the things I clarified in the learning objectives. So you don't have to be able to identify which one's mitral and which one's tricuspid. You just have to identify AV valves. The other two valves you have are the valves of blood exiting the ventricles and going into either the pulmonary artery or what's this artery called? Anybody know? The aorta, right? Okay, and those are called semilunar valves. So those are like one-way valves. So blood, as the ventricle contracts, right, blood goes out, but can't come back in. So there can be no backflow through the semilunar valves. Okay, so when you hear your heartbeat, that's actually the second beat is blood hitting these semilunar valves, right, not being able to go back in. So it would depend on the severity of it. If you have any sort of valve leakage, That's what a heart murmur is, right? And depending
Sister
on,
has a heart murmur.
yeah, depending on severity and stuff, those can be pretty benign or like something you need to.
Hers is pretty benign, but you can really hear it. And there have been a couple times where learning new doctors will come in and be like, oh my gosh, listening to her heart.
Interesting.
One doctor, when she had to go for a checkup one time, a new doctor was examining her, and he goes, "Are you aware that she has a murmur?" And my mom's like, "Oh yeah."
Yes, quite aware. My brother does too. All right. So that's the second heart sound, and that's the literal technical term for these, are heart sounds, which I love. The first heart sound is when the AV valve closes, right, and the contraction of the ventricle hits up against that valve, right? So that's the lub is hitting here, and the dub is hitting here.
It's like the majority of the pressure pushing the blood is created by the ventricle, just based on the muscle mass.
Exactly, yeah. And actually there's quite a big difference in terms of muscle mass and thickness of the two ventricular walls. So what we're seeing is the left atrium, the left ventricle, wow, it looks a lot thicker than the right ventricle. Any guesses as to why that's the case?
Pumping to more volume.
Yeah, so specifically which circuit is the left atrium, left ventricle responsible for? Yeah, the systemic circuit. Okay, so it's got to do a lot harder of a job, right? So it's got some good backup there. I guess the only other thing that I forgot to mention is that there are arteries that come right off of the aorta, which is here, right, that feed right back into the heart muscle, and those are the coronary arteries. So they're actually supplying oxygenated blood to the heart itself.
Again, those cells still need oxygen.
Right, yeah, and they need a lot of it, right? They're one of the greedier organs in terms of oxygen demand for obvious reasons. So that's not drawn here, but I just want you to be aware of that. Okay. All right, so let's ask some Socrative questions about that. First question is, what is the adaptive advantage of having a double circulation system and three-chambered heart, as found in amphibians, over the single circuit and two-chambered heart of fish? Okay, let's take a look. Sweet. Small majority in favor of B, oxygenated blood can return to the heart for additional pumping. Right? So there's a pumping between the pulmocutaneous circuit and the systemic circuit in the case of amphibians. So regardless of whether you have double circulation or single circulation, right, there are capillary beds in both the respiratory organ and body systems. So that's true for both. And this is not true of amphibians. Oxygenated blood is not kept completely separate from deoxygenated blood in the heart because they share a ventricle, right? There's only a single ventricle in amphibians. Okay, and then I kind of already stole my own thunder for this question, but why not give it another rep? Okay, which chamber of the heart do you think has the thickest walls and contracts most forcefully? All right, I guess I'll go with the first 16 of you. Sweet, okay. Pretty healthy majority in favor of the left ventricle, and both I and your heart agree. 100 out of 100 hearts agree. Okay. So the chambers of your heart, right, go through cycles of contraction and then relaxation, and that's called the cardiac cycle. Contraction, or pumping, is called systole, okay? So you can have atrial systole and ventricular systole, right? But usually when people are talking about systole and diastole, like as it relates to blood pressure, that's ventricular. Okay, and then the relaxation, right, which has the consequence of filling heart chambers usually, is called diastole. Okay, so let's sort of walk through this in a cartoon.
I remember like diastole was relaxation because when someone dies, they relax.
Oh, I like that. That's a really good one.
Or like dilate.
Yep. I don't know. I think the death one is a little more evocative, right? Okay, so let's go through a rep of our cardiac cycle. So we have the relaxation phase, okay, and during the relaxation phase, blood is allowed to flow from the atria to the ventricles. So the AV valve is open. Okay, so then just to like squeeze out that last little bit, like you're squeezing out a lemon into a tea or something, right? You do have some atrial systole, right? So there is some contraction in the atria to force the last little bit of blood into the ventricles. Okay, so the ventricles are still relaxed. They're still in ventricle diastole. Then we have ventricular contraction. Okay, so ventricular contraction is pumping blood either through the pulmonary artery or through the aorta, and blood is exiting the ventricles, going into the arteries, and the semilunar valve is preventing any backflow of that blood. Okay. So notice the time scale here, right? So this is the one cardiac cycle is really short, okay, less than a second. So your heart rate is basically the number of pumps per, usually it's minute when they measure it. And that depends on two things, right? It depends on stroke volume, which is defined as the amount of blood pumped in a single contraction, so stroke volume. And it depends on oops, sorry, I got that wrong. Cardiac output depends on stroke volume and heart rate. Okay, where cardiac output is volume pumped through systemic circuit, I'll abbreviate SC, per minute. Okay, so then your book goes into a lot of details about sort of self-excitation and all the various nodes, the pacemaker nodes in your heart. But as I was looking through the objectives, none of them were listed on the objectives, so let's leave that for the A&P courses. Okay, so here's where we talk about blood pressure. So it really is helpful while you're thinking about blood pressure to think of this exactly the way you think about like piping in your apartment or house, right? It's the same thing, right? Pretty much the same thing. Okay, so depending on what blood vessel you're talking about, arteries, veins, or capillaries, they have slightly different numbers of layers, and they're slightly different from one another. So by far the simplest is the capillary, right? All three of them have as their innermost layer endothelium. Okay, and endothelium is epithelial cells, right? Squamous epithelial cells, right? So the tiny, scaly-shaped, skinny ones that are really good at absorbing. Okay, so you have endothelium in the artery, you have endothelium in the capillary, and you have endothelium in the vein. The only other layer you have in capillaries is basal lamina. Basal lamina is not, it's extracellular, right? So it's not cells. Why might it be a good idea for capillaries not to have a lot of layers as blood vessels?
To facilitate gas exchange.
Right, because they are the site of all of these exchanges, right? So in addition to having an endothelium, right, arteries and veins also have layers of smooth muscle. And they also have layers of connective tissue. Why might it be in an artery or veins interest? Let's focus particularly on veins to have smooth muscle.
Push blood back to the heart.
Yeah, right? So like one of the ways that blood doesn't just like collect into giant pools in your feet is that your smooth muscles are contracting in your veins, and there's also one-way valves in your veins, right? So just like there are in the semilunar valves in your heart, right? So blood is slowly being contracted up, and it can't flow back down. Okay, thanks, veins. Arteries, right? Why might it be a good idea for arteries to have connective tissue? What does connective tissue do, by the way? Okay, I'm sure that plays a role there for sure. All right, so think like you're the aorta, right? And there is, right, just been a massive contraction in the left ventriculum, okay? And you have just gotten a surge of blood. You don't want to explode. So what happens? Close, kind of the opposite, right? You can actually expand a little bit, right? So you have all those elastic fibers and connective tissue, right, that prevent you from just rupturing, right? And then you can return to your original shape, right? So here, right, for arteries and for veins, right, we have some flexible piping a little bit, right? We can expand and contract because of all these connective tissues. Okay, arteries. No, vasodilation is usually in reference to arterioles, which we'll talk about in a second. Yeah. But very similar idea. Okay, so arteries tend to have thicker walls than veins, and again, it's to accommodate the high pressure. So let's look at this really cool chart that shows area velocity and pressure as blood is pumped from the aorta all the way back into the heart through the venae cavae. Okay. So let's start with area, right? Pretty small area through the aorta, right? That's not a big area. Arteries, right? You start getting slightly bigger area, not by much. Arterioles, right, which are these guys. Okay. Now you're starting to get a little bit more area. And then capillaries is where the area maxes out. Okay, so there's a lot of area for blood to go to, a lot of surface area. Okay, and then again, sort of almost completely symmetrical here as you go back from the venules to the veins to the venae cavae. Okay. For velocity, right, you start out going real quick through the aorta, and then through the arteries. And as you approach the capillary beds, right, velocity goes down, again, because surface area has gone up. So there's a direct relationship between velocity and surface area here, right, which you only need to go to your garden hose to prove to yourself or demonstrate to yourself. Okay. And then once again, right, as we're converging onto smaller surface areas, velocity goes back up, right, but it's not nearly as high in veins returning to the heart as it was in arteries leaving the heart. Okay. Blood pressure fluctuates a lot depending on whether there has recently been a contraction, right? So whether there has been relaxation or contraction, right? Whether there's been diastole or systole, respectively. Okay. Blood pressure tends to be high in the aorta and arteries, tends to be much, much, much lower in veins. Okay, so if you're taking your pulse, right, you're always doing it at arteries, not at veins.
I was told you're not actually supposed to do it at the one in your neck.
Really?
Yeah.
Oh, oops. Why?
Because apparently it can restrict blood flow to your brain, which is never a good thing. So
Just runners just collapse. Okay, good to know.
It's not that big of a deal, but it's over time.
Mm-hmm.
I think it can kind of accumulate. That's at least what I've heard from people who have studied the medical field. So
Interesting. Good to know. I feel like that should be some kind of public service announcement somewhere, right, growing up. Anyway, part of the reason you can feel a pulse, again, is because those arterial walls are expanding, right? Okay. So it's actually a good thing that velocity is at its minimum in the capillary beds, right? Because that's allowing time to facilitate gas and waste exchange. Okay. So So blood pressure, right, is the pressure that blood exerts against the walls of blood vessels, right? And blood vessels that are more rigid, right, ones that don't have this very elastic connective tissue, say capillaries, right? Blood pressure is pretty maintained, right? But in blood vessels that are allowed to deform, blood pressure can be lost. And that's not necessarily a bad thing, right? So this deformation of artery walls, right, can actually help regulate blood pressure and blood flow. Okay, so we've got blood pressure falling into two flavors, right? Systolic pressure and diastolic pressure. Systolic pressure is the pressure in arteries during ventricular systole, right? So when the chambers that are pumping blood into arteries contract, Okay, and that's when blood pressure is highest. Okay, so when someone's taking your blood pressure, right, the systolic pressure is the high number, and diastolic pressure is the low number. So that's the pressure in the arteries when the ventricles are relaxed. Okay, it is lower than systolic pressure. Okay, your blood pressure, or I guess should I say your blood pressures, right, can change by changes in cardiac output or by constriction of arterioles, which is what Miracle was getting at. So vasoconstriction and vasodilation also involve contraction of smooth muscles. They are in arterial walls. Okay, if you have vasoconstriction, that's going to increase blood pressure. If you have vasodilation, that's going to decrease it. Okay, so think about this for a second. When might you want to have vasodilation? What do you think that does to your capillary beds? Yeah, okay, so when might you want there? Okay, that's a good one. So vasodilation is relaxing those muscles, okay, bringing more blood in, which by definition is decreasing blood pressure. Exercise. Okay, that's when you need a lot of O2 exchange in your capillary beds, right? Because you're using up a lot of O2 to digest or to exercise. Okay, so just looking at this, you might predict, okay, if there's a lot of vasodilation during exercise, shouldn't blood pressure drop? And the answer is no. But my question to you is why not? Carlos. Yeah, right? It's because your heart rate is going up, right? So your heart rate is increasing to keep blood pressure stable, okay? So your blood pressure isn't changing a lot. It's just your cardiac output that's changing a lot. Okay, let's take a 5-minute break, and I'll see you guys at 6:05. All right, it's 6:05. Let's start back up. So the last two things I wanted to say about vasoconstriction and vasodilation is that various things can induce both of these. So the main one that's responsible for vasodilation is nitric oxide, not to be confused with nitrous oxide. So we're talking NO, not NO2, the thing you get at the dentist's office, right? NO is what causes, vasodilation. And the peptide that causes vasoconstriction is endothelin, okay, which is spelled like this. Okay, so your body can regulate vasoconstriction and vasodilation using nitric oxide and endothelin. Okay, can couple those with heart rate and cardiac output in general, right, to regulate blood pressure. All right, so now we'll ask a couple more Socratic questions. Feel free to discuss with partners, friends, neighbors. Okay, let's take a look. All right, so the answer is false, right? Most people said false, and that is correct. So, right, for example, we can increase blood flow by vasodilation alone, right? And if we wanted to maintain blood pressure while doing that, all we would have to do is increase heart rate, which is exactly what happens when you're digesting or exercising. Okay. By the way, one of the reasons they don't measure, basal metabolic rate after you've just eaten a big meal. Okay, so now let's talk a little bit more about how they measure blood pressure. Okay, so when you go to the doctor's office, right, they always measure blood pressure, right, and they do it by wrapping a cuff, usually around one of your arms, and they pump that cuff, right, and they know the pressure that they're applying to the cuff. It's usually measured in millimeters of mercury, okay, for historical, chemically dangerous reasons, and then so they wait until. So they usually have like a stethoscope or something downstream of the cuff, and they wait until it's completely constricted blood flow. Okay, so that means the pressure of the cuff is now greater than the pressure of the artery, right? Because the artery can't apply enough pressure to continue supplying blood to your arm. Okay, so then what they do is they relax the cuff until they can start hearing blood flow happen. Okay, so the point at which they can, yeah, exactly. So the point at which they can start hearing blood flow is your systolic pressure. And they continue relaxing the pressure in the cuff until they stop hearing anything at all. Okay, and that's your diastolic pressure. Okay.
So
Yeah.
why would they stop hearing things that they're measuring if they're still using the...
Not clear to me. I had like the same question written in my textbook, so I'll try to find out since you have the same question.
I'm assuming that the sounds that they're hearing are the blood as it,
Like returning?
as it starts hitting against the vessel walls again, and then once it's come back and it started flowing again, you can't really hear anything because it's just continuous.
But you can feel it in your pulse, so that's what confuses me.
Right. Yeah. Book does not say, that's for sure. Okay, so it wouldn't be a test question, but I'm also curious about that.
Maybe the actual something is just the blood rushing into the vessel. And once that stops,
Yeah, but even if you had like partially constricted, presumably that would eventually stop, right? I don't know. For those of you who are shadowing medical doctors, this would be a good question to ask your medical doctors for your community service or service learning. Okay, so then your book kind of hilariously talks about fainting as like an adaptive trait, which it kind of is, right? It kind of makes sense, right? So, well, in some cases it's an adaptive trait. So the idea is your brain, if it's not getting enough oxygen, right? It has to put some effort to pump oxygen-rich blood all the way up to like this high part of your body, right? And it's like way worse if you're a giraffe, by the way, right? So like blood pressure in giraffe necks is like insanely high, right? Because it has to work really hard to get that far up. So if at any point, right, blood pressure kind of drops, right, and the brain isn't getting enough oxygen, it will cause you to fall over so that you're horizontal with your heart, and blood can return to your brain.
Is that why it's harder to sleep if you're sitting up rather than laying down?
I don't know about that. That's a good question.
Because your body needs to work harder to pump up and around your body rather than
Maybe. I wonder.
horizontally, so you can relax
I wonder.
go to sleep.
I wonder if you lose more weight falling asleep on on a chair than if you do.
It's just like more strain on your muscles.
Yeah.
Because it's like you have to like maintain your posture, so it's like that's putting pressure on your neck muscles and your back muscles. So it's like I think it would just be easier for them to lay down.
Yeah. Yeah, so by the way, speaking of giraffes, your book goes on to talk about giraffes, and they when they bend down to drink, right, blood pressure is so high in their necks that like it would cause brain damage, right, if their blood pressure stayed that high while they bent over to drink. So they have all these like adaptations that like regulate blood flow while they're bending over to drink, so they don't die, which is kind of funny. Yeah, so your book actually also talks about that, right? And it talks about this hypothesis that some of the really, really long-necked sauropods probably didn't like forage on the treetops because there's like they can't think of a way to physically explain how blood pressure, like how there would be a high enough blood pressure for that to be possible. So what they think is that they foraged at like I know, right? It seems.
They were like worms.
Yeah, just like worms with giant bodies, just like walking behind them. Yeah, so I'm sure that there's something that we don't understand because it just seems like a very unlikely hypothesis. But your textbook was like, this is what people think, so.
I mean, it apparently worked, whatever they did, because there were a lot of
Yeah, they were pretty successful in their time. Okay, so it's not just smooth muscle contraction that enables your veins or blood to return to your heart from your veins. So skeletal muscle contraction is also involved in this. So as you're walking around, right, you're actually facilitating returning blood to your heart.
This is why you can't sit down and play video games for 5 days straight.
Also, why it's hard to just stand in the same place for a really long time. Yeah. I think so. Your book doesn't mention that, but that was like my first thought when I read the book. Is I was like, I bet that's why. So don't quote me on that, but.
You're right.
Am I right?
Yeah, I remember when Archie was saying something like that's why it's not good that cashiers stand so that the skeletal muscles can contract.
That's like, isn't that like varicose veins, like with like waiters and stuff like that? Or is that a different?
With what?
Like waiters and waitresses that have been doing the job for so long, you know, they get varicose veins and they're like,
I don't know.
related to being alcohol.
Yeah. That's a good question. I mean, if that's like an occupational hazard that's known for servers, then yeah, I bet it's related to that, but I don't know the physiology of why that would happen. Okay, at any given time, right, about 5 to 10% of your body's capillaries have blood flowing through them. But at almost all times, blood is flowing through the capillaries in your brain, your kidneys, and your liver, and your heart. So blood flow is regulated in these capillary beds by two mechanisms. So vasodilation and vasoconstriction, which we already know. And then these are called precapillary sphincters. And they also control blood flow between arterioles and venules. Okay, so now in the coming slide, we will talk about how gas and waste exchange actually occurs in the capillary beds. Okay, so the good news is both oxygen and carbon dioxide can diffuse via simple diffusion. right? They don't have to be carried through any sort of channel proteins whatsoever. They can just very easily go through plasma membranes. But blood vessels actually do have pores that allow there to be easier gas exchange between for CO2 and O2, as well as a bunch of salts. Okay, so big blood proteins can't really go through these pores, and neither, of course, can erythrocytes and leukocytes in big cells. What else did I want to say about this? Yeah.
So it's only the O2 and different nutrients that are flowing out. It's not actually like the whole cell, right? It's not near big enough, the little pores. They're just going by, like through the capillaries. O2 is being released through the pores into the interstitial fluid, right?
Right.
And then, is there any reason that the arrows are like growing?
Yeah, that's what so like when I practiced this earlier, I had this slide down, and I like totally lost my train of thought. So I'm like looking at my notes here. Oh yeah, it's in the plot below, right? So yeah, what's happening is as it's going across the capillary bed, on the arteriolar side, blood pressure is high, right? Because it's coming from an artery. But as blood pressure goes down, so the flow of products from the blood into the interstitial fluid depends both on osmolarity differences and on pressure, right? So if there's enough pressure, you can push things against their concentration gradient, right? So and that can be the case on this left-hand side, right? But as blood pressure goes down more onto the venous or venous side, blood pressure is so low on that side that actually facilitates diffusion of things back into the blood vessels.
And then the blood pressure, does it grow when things are added again? Like,
I don't think, right, because none of it's fluid, right? So it's all stuff, yeah. So I don't think so. Yeah, let me check. Does that answer your original question or no? Okay. Let me check my notes and see if there's anything else I wanted to say.
Do we know which one comes out and which one comes in?
I mean, so presumably CO2 is coming out. Well, it depends. Yeah, it depends on where it is. Yeah. Let me just read my notes. Give me a second. Stop asking me questions. No, I just didn't remember what I wanted to say. Yeah, I mean, I think it's going to depend on whether you're in the pulmonary circuit or in the systemic circuit. Right? In the systemic circuit, you're going to want oxygen to diffuse out, CO2 to diffuse in. Right? But if you're in the pulmonary circuit, you want oxygen to diffuse in and CO2 to diffuse out. So how specifically those things are regulated, I think, depends on concentration inside the blood vessel. Right? So if you're in the pulmonary circuit, right, the concentration of O2 is probably really high out here, and it's probably really low in here. So O2 is going to flow in, and CO2 is probably higher here than it is here, so CO2 is going to flow out. Does that help? Okay. I had no helpful notes for that. Okay. So you do actually lose some fluid in these exchanges as well. And in fact, you lose an average of 4 to 8 liters of fluid a day from your capillaries. So how that affects blood pressure is a really good question. I'm not quite sure. But the thing that essentially recaptures this lost fluid, and actually also some lost blood proteins. So I did mention that blood proteins have a really hard time going through, but they do occasionally get out of your blood vessels. And your lymphatic system recovers all of that. So your lymphatic system also plays a role in immunity, which unfortunately is not something we are going to talk about in this course, which I'm super bummed about because I was really pumped about learning about the immune system, but we're not. Yeah, that's chapter 43. Now I'm like super bummed. But the lymphatic system also does play a role in recovering a lot of these materials, right? So that fluid, which is called lymph, right, does re-enter the circulatory system at veins in the neck. So do trust me on that one now, right? It's definitely official. Okay.
Those are the lymph nodes.
Yeah, so lymph nodes, I'm so glad you asked. Okay, are organs that filter lymph. Okay, so they basically are like honeycombs of connective tissue, and there's a bunch of white blood cells inside them. So when you have an infection, your white blood cells will proliferate, right? And that's why your lymph nodes get swollen when you're sick. That's why your doctor checks your lymph nodes. And also, it's something that they check to see whether cancer has metastasized, right? So metastatic cancer is when cancer leaves a particular organ system or organ and proliferates to other parts of the body, right? So it needs to use some sort of circulatory mechanism to do that. So cancer cells are actually sequestered in lymph nodes, so they can actually tell whether, or use that as an indicator of whether cancer has metastasized as well. Okay. Okay, so now we'll talk about blood composition. Very much the like 30,000 foot view. We're not going to jump into the details of all the various leukocytes. That's something that you're not responsible for. So it's worth repeating, right, that blood is connective tissue, right? So blood is just very specialized connective tissue. And if you were to take blood and centrifuge it out, it would filter into more layers than this cartoon is showing. So it's funny, this is the older edition of your textbook. The newer edition has a third layer, but there's actually more than 3 layers. So the top layer is plasma, blood plasma, right? And inside blood plasma, you've got your water, you have all your various ions, and they're involved in osmotic balance, right? So osmoregulation, which chapter 44 talks a ton about. Buffering, regulating membrane permeability. It also has a bunch of proteins, right, involved in clotting and defense. And it also has nutrients, gases that are transported. Okay. The layer missing from this figure, but present in the new edition of your textbook, is a layer between these two called the Buffy Coat. And the Buffy Coat contains the leukocytes and the platelets. Okay, so leukocytes are all white blood cells. White blood cells have different tasks. And then platelets are fragments of cells, and they're involved in clotting. And then the final layer is the red blood cell layer. And surprise, surprise, it contains red blood cells. Okay? Red blood cells are also called erythrocytes. And we'll talk more about them in the next slide or so. Okay, so the red blood cell layer and the Buffy Coat layer comprise about 45% of the cell volume, and the remaining 55% is plasma. Okay, so before you centrifuge these out, right, so we just separated them by density. That's what centrifugation does. They're all suspended, right? So all these cells and all of those ions and nutrients are all suspended in the blood plasma, right? So that's just a liquid matrix of blood. Nothing new on this slide, right? You know that red blood cells transport oxygen. You know that white blood cells function in defense, or hopefully you know, maybe you don't. That's okay, either way. And then platelets are involved in clotting. Okay, erythrocytes specifically are by far the most numerous blood cells. They are enucleate, right? So they do not have a nucleus. They do not have mitochondria. Okay, so they are doing anaerobic respiration, which is kind of ironic because they're oxygen transporting, right? But they're not doing any cellular respiration, aerobic cellular respiration themselves. So basically, they've removed everything like to accommodate hemoglobin, right? That's pretty much all they do. If you look at the shape of an erythrocyte, even the shape of the erythrocyte is optimized so that there's a lot of surface area that allows diffusion of oxygen into red blood cells. Okay, a single erythrocyte can accommodate, I think it's like 250 million, is that right? Yeah, molecules of hemoglobin. Okay, each molecule of hemoglobin can accommodate 4 oxygen molecules. So a single erythrocyte can house 1 billion oxygen molecules. So that's a lot of oxygen per erythrocyte. There's a lot of erythrocytes. Okay. So hemoglobin is that pigmented molecule, right, that we were talking about. And we know from previous courses, right, that hemoglobin has a high affinity for oxygen. And that affinity changes based on cooperativity. Does anybody remember cooperativity? What's that about? Cooperativity. Really? No one?
When oxygen comes in, it has a higher affinity to collect more.
Exactly. Yeah, right? So the more oxygens that are bound, the more oxygen molecules that are bound to hemoglobin, it actually, every time oxygen binds, it changes the conformation of the hemoglobin just a little bit and makes it have a higher affinity for the next oxygen, and so on and so forth. So it's ringing a bell now to like no one? Cooperativity we did last quarter. Yeah, totally.
Last quarter.
Yeah.
Oh.
Oh, I'm sorry. Knowledge gets thrown out the window every time. My bad.
Way back when.
Okay. Oh, I don't know if that speaks to your learning or my teaching worse. Yeah, I think it speaks to the way college works more than anything. What were you going to say? Sorry.
No, I was just thinking about last week. I was like, did we say that last week?
No, I did not.
Yeah,
I did
that's what
not.
I was thinking.
Yeah, months and months ago. Yeah, I don't even remember what I had for lunch. I shouldn't judge you guys. So, anyway, okay. But all right. So cooperativity. Are we now somewhat familiar? Right. It's this increase in binding affinity. The more oxygen you have bound to hemoglobin. And then pH also affects hemoglobin's affinity for oxygen, as we will talk about if we get to that part today. Okay. Leukocytes. You don't have to know the names of any of these words, right? You just have to know. Some leukocytes phagocytize, right? So eat in the cellular sense, right? Bacteria or other debris in your bloodstream, and other leukocytes produce antibodies. That's all you have to know, just those two things. Okay, so platelets are involved in clotting. And they're actually kind of involved in clotting in sort of two different ways. So let's walk through this. Let's say that, so again, platelets are not full cells, they're fragments. So let's say that you're in a blood vessel, right? You've got these big old erythrocytes hanging out, and you also have platelets, and something happens that causes damage to the vessel. Okay, so what's the innermost layer of a blood vessel? Connor's got, I think, most of it. Endothelium, right? So it's a pretty thin layer, right? It's just one layer of cells. And what's beneath that layer? Smooth muscle. Okay, what's beneath that layer? Connective tissue, right? So once platelets get exposed to collagen fibers in the connective tissue, they will bind to those. Okay? So they'll basically just accumulate around those exposed collagen fibers and just kind of quickly patch up the damage, right? So that's not a long-term fix. So when platelets bind to collagen, they actually will release compounds that make platelets near them sticky and will stick to them. Okay, for some reason they didn't label it until later.
Is it always collagen that triggers it?
Is it always what?
Collagen that triggers it?
I believe so. I believe so. Okay, so now we've got our platelet plug.
And they're just always present in the bloodstream.
Yeah, so they're constantly circulating, as are a bunch of other clotting factors in the blood. Okay, so for example, prothrombin is a protein that's in its inactive form, and it's just flowing through the bloodstream all the time. But when these platelets clot, they'll start an enzymatic cascade that ultimately leads into conversion of prothrombin into thrombin, which is the active form of the protein. Okay, so both your book and I are being very vague about that particular enzymatic cascade. And the only part that they and I want you to care about is this conversion of prothrombin to thrombin, and then thrombin acts on fibrinogen and activates it into fibrin. So fibrin is what forms like a stronger clot, like an actual blood clot, right? It's just this interwoven network of fibrin threads, and they can tangle up erythrocytes, right? And that's when you've got a pretty good seal, right? A pretty good clot on damaged vessels. Okay. So as you might imagine, this is a good thing for blood vessels because it's preventing, right, like hemorrhages. But it can also be a bad thing, right? Once clots get dislodged, you can have cardiac issues, stroke, etc. Okay, so grab a piece of paper and a partner, write your names on it for participation. Just try to outline those sequence of events that occur during blood clotting. Spencer go to weigh CO2? Is that what he's doing right now?
He had to work
He's
away
a duck
from
out.
off the same pretty fast.
For damaged artery. And then the platelets will release the chemical for nearby platelets into and then all this somehow is Prothrombin into Thrombin. A series of
So the platelet, well, first we have vasoconstriction. Process of prothrombin.
And then they produce
Which then I guess like catches fibrin, it's like a net that catches.
and then you drop the milk down. And it's just fibrin.
Sure, I'll take it. Thank you. Does your whole book come hole punch like that? That's awesome. Is that how they sell it or not? Oh, okay. That's brilliant. That's awesome. I'll do that. Huh. Okay, raise your hand if you're still working on this. Couple folks. All right. Give you like another minute or two.
I think it's a whole bunch of things that can lead those things for a while.
That's awesome.
Are you gonna write like
Okay, in the interest of time, since I only have 10 minutes and I want to equip you with as much as I possibly can before you have to study it independently, I'm just going to surge ahead. So, all right, next we're going to talk about gas exchange and respiratory surfaces. Okay, so the first objective that this section of your learning objectives has is it wants you to be able to distinguish between respiratory surfaces and respiratory media, right? A respiratory medium is something like air or water, right? It is what the oxygen is found in, okay? So, surprise, surprise, per volume, right? Water is always going to have less oxygen than air, okay? So obtaining oxygen from water is always going to be less efficient than obtaining oxygen from air. Therefore, it's going to be harder and require more energy. Okay. So for example, fishes and lobster, right, they have to expend a ton of energy just to carry out gas exchange. Okay, so that's respiratory media. Respiratory surfaces are always moist, by the way, right? So they always have to be aqueous to allow plasma membranes, right, to incorporate or diffuse out these gases. So that's even true for terrestrial organisms like us, right? Our respiratory surfaces, right, the alveoli in our lungs are wet. Okay. So
are they covered in mucus? Or is it just like water?
I think it's just fluid. Yeah, that would probably not be good.
How does the water not cool? I guess like
So yeah, might as well, because it's a question in the learning objective anyway, right? So your alveoli, if there's fluid around them, that can cause surface tension. And that surface tension can collapse your alveoli, which is why you have a surfactant, right? Yeah, in your alveoli that reduces surface tension, right? But that doesn't happen during infancy until like a couple of months into gestation. So if you have a premature baby born, if it weighs like less than 2 pounds or something, they have to give it like a substitute surfactant so it can breathe on its own, right? So that was like a really cool medical breakthrough that they discovered. They were just like, oh, if you have a really premature baby, you have to administer this surfactant before it can start making its own. Otherwise, its alveoli will collapse. Okay, so that's what it's talking about when it talks about the surfactant in your learning objectives. Okay, various different kinds of respiratory surfaces. I don't think there's an objective that goes into the specifics of that, so I hope you'll forgive me for moving on. Okay, oh, thanks. All right, so ventilation is the word we use to describe the process of moving respiratory media over respiratory surfaces. Okay, so this can happen while you're locomoting, right, which is something that happens a lot with fish, sharks, et cetera, right? They have to actually locomote to ventilate, right? By the way, not all sharks. Not true for all sharks. Some sharks can just be fine being still. They can still breathe. Okay, common urban myth. Okay, so fish in particular, they actually use countercurrent exchange while they are doing this, while they're ventilating. Okay, so we've seen countercurrent exchange before when we talked about thermoregulation, right, in those limb extremities. And here's another example of countercurrent exchange, but this one has to do with increasing the efficiency of gas exchange. Okay, so let's take a look at this. Okay, so you're a fish, you're taking in oxygen-rich, right, water through your mouth, right, and you are flexing your operculum, which is your gill cover, right, and both of those actions are pumping water through from here, so oxygen-rich to oxygen-poor, okay? So here is a gill arch. There are several gill arches, right? Each gill arch has blood vessels, okay? And those blood vessels are organized along these gill filaments in such a way that you always have the most oxygen-rich blood encountering the most oxygen-rich water first. Okay? So the oxygen-richest water is still going to exchange oxygen with the oxygen-richest blood. Okay? If you had oxygen-poor water, it wouldn't be able to diffuse oxygen onto the oxygen-rich blood. Did I misspeak?
Why not? Is it too dilute? Or?
It's too dilute, right? So if you look at the partial pressure of oxygen here, if you like absorbed oxygen earlier, and then you want to absorb more oxygen, you would have a partial pressure of 30 in the water and a partial pressure of 140 in the blood, and it would never happen. You would actually diffuse oxygen out of the blood.
Oh, I see. It's about.
Yeah. So basically, as you're absorbing oxygen, the oxygen concentration in the water is going down. Okay? So you have the oxygen poorest blood, right, being sort of the last part that gets exchanged with the water, because it's the only part that's low enough to still accept oxygen. Okay. All right, there was so much more I wanted to talk about. I guess I'll make a quick video, and I'll post that probably tonight with the rest of the material. I'll collect your participation, and if you haven't gotten a Scantron for exam 1 yet, come see me.